So thank you for the invitation. I'm professor at Polytechnique for 35 years, and I don't remember the time that I came here to give a seminar. So maybe it's my first time that I'm giving a seminar at McGill. While I'm collaborating with all of the people at McGill, it's my first time. So thanks to Dan for the invitation. Sometimes I give talks everywhere in the world, and never thought to come to McGill. I mean, I go to many universities, but yes. What I'm going to talk about is the application of physics. I'm a physicist. You will see from what I will be talking about is the application of physics, and mostly application of using optics in the field of biomedical. And essentially, I look at the problem from the physics point of view, and then move to the field of biomedical mostly. Before going to that, I've been told not to use my pointer, so it's very difficult for me because I usually use pointers. I'm part of the Canada First. This is a big project. This is $35 million that we receive on the application of engineering to fabricate various types of devices, therapy, diagnostics for the medical field. It's a transdisciplinary, intersectorial open innovation ecosystem for the needs of innovation product implemented in the health system. So probably I'm going to use my hands here. That would be better, okay, for the recording. So the mission of the TransMedTech is to develop and validate the next generation of medical technologies for important complex diseases, as well as to transfer and implement them efficiently in the health system. And so this is a very important grant. We're looking for students, actually looking also for higher research personnel and a professor in this field. So basically, we cover, this is the only slide in French. We cover mostly illnesses in musculoskeletal, cardiovascular, and cancer. And that covers a lot of various illnesses in the, medical problems. And we approach that with diagnostics, which is basically the approach of nanotechnology, imaging, various medical devices, and also some technology to readapt. So this is mostly the field of my colleague, Carl-Éric Aubin, who is the head of this institute. This is the Institute TransMedTech. And so there are university and hospital, and part of this part here that is McGill. This is the Jewish General Hospital. So it's Polytechnique with University of Montreal and various hospitals around the university, and many other partners. Okay, so this was only for the advertisement. So that's my group. So I'm a physics background, mostly materials and optics. So I use materials and optics. So basically, we develop ultrafast laser processes, nanomaterials for biomedical, bioplasmonics. I will explain what is plasmonics in a minute. Nanotherapy, nanodiagnostics. And basically, we have an infrastructure of close to multi-million dollars equipment of various lasers, various plasmonic systems, spectroscopy, cell culture, and also we're starting to have in vivo facilities in our lab, but usually I do in vivo in collaboration with my colleague at the CHUM or Saint-Justin, or HMR is Hôpital Maisonneuve-Rosemont. So this is basically that. So basically, my team is composed of physicists, engineer in physics, engineer also in biomedical, and also biologists, and also linked with other person and other researcher in hospital, also medical doctor. So we look at more of the physics side of plasmonics, nanoplasmonic imaging, ultrafast lasers. This is what I'm going to talk about. This is more the engineering development, where we develop plasmonic devices, nanomaterials that I'm going to present. At the same time, then developing systems. Nanosurgery, I'm going to talk about this, drug delivery, and also the application. This is what I'm going to cover today. Two applications, ophthalmology and pathology. These are the two types of application of what we are developing. Okay, so this is the general introduction. So I'm going to talk two topics. One is probably 2/3, and the other one is maybe 1/3. That's basically the time frame. And I'm going to introduce to you and explain to you a new technique to perform surgery in living cells on a nanometer scale. And so we'll start by explaining where I come from, why I'm doing that. And right now we're doing in vivo application in the eye of animal model. But basically, we started from quite a long way to arrive to there. And I think it's, personally, I think it's very interesting and challenging. And that's another one. This is for bioimaging. And basically, this is mostly for pathology application. The idea is to use these nanomaterials, plasmonic nanomaterials that I'm developing, and to help pathologists. And I'm working with pathologists. And on this one here, I'm working with directly with ophthalmologists, and so to apply this for the surgery directly in the eye of cells. Okay, let's start with the first topic, and I like history, so I started very far from now. Okay, history of surgery. This is a book that I think it's only in French, but probably it will be in other languages. This is the history of surgery, and basically in the history, I mean, we started very far here, 10,000 years ago. This is the 13th century in the Middle Age, and right now the scalpel that is quite heavily used, of course. The scalpel has some limitation in terms of surgery. It's mostly this angle here that has the limitation in terms of what we can perform in terms of surgery. We can use a laser. Okay, that's my specialty. So you use laser to perform surgery in cells. So the idea is that surgery are being around for quite a number of decades now. It is used for various types of application, and probably one that is well known is to do the surgery in the eye so that you can have eye corrections, in which I think it's very common now. It is limited in terms of dimension. This is not necessarily nano. I mean, it's much larger in terms of its dimension. As you probably know from your basic optics course, undergraduate course, don't look at the equation. This is not pretty particularly important for the moment. It's just that when you focus with a lens, you arrive with dimension that will be on the order of the wavelength. So the wavelength of in the visible is around half a micron, 500 nanometer, let's say, half a micron. So this is the limitation. Actually, it's the limitation, optical limitation, but the actual phenomena is due to the fact that when you irradiate with a laser, you have a lot of dissipation of the energy. And actually, this region here is much larger, actually, than half a micron. So it's many microns, actually. It's sufficient for many things. So it's many applications. The question is, can we do better? Okay, if we cannot do better, we will not be here. So we'll proposed to go with the reference to this book here of the history of surgery. The great surgeon of the future will be a nano surgeon. So being able to perform surgery on a nanoscale with different approaches. I'm not saying that the one that I would be proposing is for some type application, but of course, there may be some different other ways. What I'm going to use is an ultrafast laser. So it's a laser that has a pulse width that is sufficiently small that you will get many types of phenomena on a very short distances and limits the irradiation. So that's the use of this type of laser. So femtosecond laser here should not be here. That's I agree. Yes, this is the mix between French and English. It's to limit the energy diffusion, limit the heat affected zone, and essentially it's based on nonlinear phenomena, which is pretty interesting. In fact, I mean, this type of laser started with some work that was done by Gérard Mourou and Donna Sticklin that just got the Nobel Prize a year ago. And Donna Sticklin is a physicist at Waterloo University. And this is Canada. So we received yesterday, no, 2 days ago. 2 days ago, she was at Polytechnique giving a talk. And we had an interesting discussion together. So she is the inventor of this. And so this is a very, very important tool in various fields, including biomedical. This is a tool that can do many things in imaging and also therapy and in this nanosurgery. So maybe a little bit of physics. This is what it looks like, an ultrafast laser, by the way. I mean, this is a laser, of course, this is a solid-state laser. But I mean, a laser is much bigger. And actually, a laser like this, it's no longer something like you have an amplification and two mirrors. I mean, you have a lot of other mirrors and dispersive materials and so on. And so this is an open box ultrafast laser, 800 nanometer, 100 femtosecond in this case, 100 femtosecond. This is very, very short. The oscillation in a molecule is picosecond, so it's smaller than an oscillation in molecules. So this is pretty short. That gives rise to many types of phenomena. One thing that happened that was used, this is almost 25 years ago, is that if you try to do this with a nanosecond and this is with a femtosecond, I mean, that's very much cleaner in terms of machining. This is stainless steel. This is not biomedical, but the idea is maybe to move to biomedical. So the idea here is to instead of having a one photon absorption, in a femtosecond time frame, there's so short in time that you can have many photons absorbed at the same time. And this is through virtual states, so multi-photon absorption. And this is one of the basic principles that you can do in terms of in terms of absorption and so on. And the idea is through an irradiation. So if this is the material and this is the beam that is focused, it goes like this. At this plane here, you will get like an intensity that looks like something like a Gaussian here. And this is quite standard in optics. However, if you have multiple photons, this is the region. See, if you have multiple photons, if it is square here, this Gaussian is square. And then the region is smaller. And then third, the region is smaller, and then if I continue, and so on, and what you get is a region that is much smaller. So in this case, I can do surgery on a nanoscale, and maybe an application, two applications from some of my colleagues that I know, Eric Mazur at Harvard and Karsten Koenig in Germany. We can break here very fine microtubule, for instance, or something in a cell, and without killing the the cell. So you can do like micromechanics or the mechanics of cells and all that stuff. A lot of, this is a tool for that. But this is still big. Okay, hundreds of nanometer. Can we do better? Okay, again, if I cannot do better, I will not be here. So we move to the use of femtosecond laser and nanotechnology. So we're going to couple these two, I mean extreme, femto in terms of time, and nano here in terms of distances here. So not only we can do better, but we can do more. So this is what we started to do some years ago. So an introduction a little bit on plasmonic nanostructure or plasmonic nanoparticles. So plasmonic is that here. Yep, I brought that. Okay, here. These are gold nanoparticles. Gold nanoparticles in a liquid. Okay, it is red. It is red because I am going to explain in a few seconds. See, so these particles are metallic and are small. Here it's around 13, 15 nanometer, maybe a little bit larger. We will go to up to 50, 60, 100 nanometer in some cases. Colors changes with dimension and also composition. This can be made in few seconds, in few minutes. If you take my graduate course at Polytechnique, you would make that in the lab. I have a course on biomedical nanotechnology. So this is called plasmon. It's a collective free electron oscillation in a metal, and basically it gives rise to this type of absorption phenomena, scattering, and absorption is mostly in the visible. 500, 600, this is visible. So particles like this absorbs in the visible. What is left is here. This is the red. So basically a solution like that looks red because this is what it is not absorbed. It reds due to that. So basically this is this part here. So these nanoparticles are also some specific aspect that when it is irradiated at 800 nanometer, there is a field amplification. This is the field as a function of the space. And here, the field is 4 or 5 times, maybe sometimes larger than that. And so there is a concentration of energy. And so the idea here is this field amplification can induce highly localized processes on the nanoscale. So this is an example of this is a nanorod, but the example is so the idea is that if this is a nanoparticle, okay? Pretend it's a nanoparticle. Okay, if I put a nanoparticle on a surface here and I irradiate very large, not focus, I irradiate everywhere. Around a nanoparticle, there will be a field amplification, a concentration of energy, and at some point, it creates a hole there. This would create a nano hole. The hole can be tens of nanometers, something like that. Can be very, very small. This is a piece of silicon, and this was 10 years ago. 10 years ago, I was putting nanoparticles, my student, yeah, not me, putting nanoparticles on the surface, irradiate, and find that there was a little hole. You say, oh, that looks interesting. So why don't we change substrate? And that's why I'm here, because this is bio. We changed for a biological media. So we're going to put these nanomaterials on a cell. So this is nanoparticle, put it there. And when I irradiate, this will create a little here surgery, whatever, process on a nanoscale, very close to the particle. And this will concentrate the energy. And the particle does not break, by the way. I will show you that. This is basic from the basic physics. It is not broken. So we can apply that to biology, and I call that the multi-nano scalpel. It's a scalpel, but at many places, because I'm not putting one particle at a time, I will put many. I will put many at many places. And that's one idea. So why do I use ultrafast lasers? I use this ultrafast laser at 800 nanometer Because at 800 nanometer, the absorption is very small. Absorption is very small. In the visible, where biology and so on absorbs quite a lot. At 800 nanometer, absorption is the light can penetrate, let's say, centimeter or many centimeters. So if I have cells everywhere here, I have cells, tissue, and I put one nanoparticle there, I irradiate at 800 nanometer. Irradiation at 800 nanometer, depending on the energy, of course, but if I don't put too much energy, I just put just enough energy to be amplified here and not there, I will produce something here and not there. And that's the main idea. So these are cells drawn by a physicist, of course. Okay, they're not so nice. Okay, so if I focus a normal laser, I would produce this. So this is like microsurgery. But if I put a nanoparticle there, I can irradiate. The beam is not focused, and I will produce something that is very small. So this concentration of energy is due to the particle. Okay, so I can also functionalize. So maybe you're certainly quite more expert than me as a physicist, but in my team I have biochemists that are doing that. So on nanoparticle, I can put some kind of biochemistry, bioconjugation, so that this will recognize, like a seeking device, will recognize different parts of the cells, cancer cells or whatever. Okay? So the idea is that, so if I want to do multiple nanoscalpel at targeted places, let's say I want to target these and not the other ones, I'll have to recognize some targets on the cells. So the biologists and the biochemists tell me what to put on the particle, and then I can do this nanoscalpel. So the idea is that I can put these nanoparticles, do that, and produce multiple nanoscalpel. So this is nice. So what can I do with that? Now I have a tool now. I have a new scalpel. This is a combined use of a laser and also nanoparticles. So what can I do? I can do many things. So from the physics point of view, I come back from the physics background now. It is an interesting, a very interesting aspect of nano and femto. As a physicist, this is great. This is interesting because you have many phenomena that you don't get if you are, in like a normal life with centimeter and seconds here. So the time is very short and distances are very small. What I can do is that if it absorbs energy, this becomes hot. It will transfer the energy around, and the surrounding can become hot. Biology is mostly water. So in all that what we do, we say it looks water. We can also create hot electrons because of this near field. So it's like a hot plasma. It's like electronic plasma, not plasma for biologists, electronic plasma that is produced close to the particle. And this will probably get to the point that the water would be heating. And then the release of that can create some kind of a small pressure wave, a pressure wave that is like a tsunami. It's like you produce something and then you have this wave coming out. But you have to consider that this is nanometer scale. And then can generate a bubble. And this is something that we have measured. Maybe we can measure these bubbles. Okay? So the idea is that it is acting like a nano lens. So it is concentrating the energy on a very small scale. So it can be something like a nano heater producing nanoplasma, a nano tsunami, or some kind of nano bubbles. Okay, This is for the physicists, okay, and this is the time scale, and we have understood that, so I'm going to skip that, but this is very interesting. My PhD student that did that won the best PhD at Polytechnique 5-6 years ago. The student was brilliant, but the topic was also pretty challenging and leading to all what we are developing today. So basically, so these are, I have to go back here. So these are the bubbles that we can measure. So they are real, okay? This is, we have done various optical setup and so on. So we can produce bubbles. These bubbles are much longer than the pulse width. They are nanosecond time frame. I mean, tens or 100 nanosecond. And so these bubbles, something that I can measure. Now, what can I do with the bubbles? But the first thing I'd like to answer is the particles are not broken. The particles are not broken because this is important. If we irradiate at 800 nanometer, the absorption is very, very small. So there is no energy absorbed in the particle. However, the scattering, which is this here, this near field leads to the generation of all the energy and so on. So and we have checked and everything and looked, but particles do not break. And this is, it's like a scalpel. A scalpel, you don't want to use a scalpel and then you break your scalpel. So you're not breaking the nanoparticles. So if you did not follow all the details about the physics that what I just explained, you just look at this page here. This nanoparticle that is here and that is here is concentrating the energy like a nano lens. So concentrating the energy at one point, actually at 2 points, here, because you need to solve all the electromagnetic equations and so on, Maxwell's equation, but basically that's the result. So what we can do with that, we can use some of the energy to kill cancer cells or highly localize. We can produce enough electrons to do different types of processes, like creating like a reactive oxygen species and so on. I mean, create some kind of other species with highly energetic electrons to create specific processes. We can produce a pressure wave, like if you do photoacoustic imaging, you can have some nanoparticles there that will enhance the phenomena. And so this can be like a high contrast, or you can produce a bubble. So the example I'm going to show now, we're going to emphasize, is this bubble. The bubble will create pores on membranes, and what I'm going to talk about is like electroporation. So who knows what is electroporation? Usually in biomedical, you should know what it is. I'm a physicist. I didn't know before I started on that. So electroporation essentially is to create pores so that you can do gene transfer and incorporate other molecules or the biological molecules into the cell. So basically, it's the following. The idea is to do transfection, probably a topic that you have seen in some of the biological courses. So if you want to include some foreign DNA, you can use, approaches like using liposome, or using viruses, or using more physical techniques. And these physical techniques can be an electrical shock, like electroporation. So you put the cells and you put them in a needle and you produce electrical shock. You kill a lot of cells, and then some other you can include what you want to include. And you can use laser also. And what I'm introducing is to use laser, but to do targeted optoporation. So this is transfection. So this is a cell, foreign DNA that you want to get in, and basically you create pores into the membrane, and then you can include what you want. So this is a little video about what we planned. This is a video animation. This is not the real. This is an animation. So you have a gold particle. These gold particles that we put, this is not a snowstorm. This is particles going on the surface of the cells. And then we have some kind of plasmid or whatever that you want to include. And that's the way of trying to do this transfection. Okay, so we put these nanoparticles, they can be functionalized, and then one pulse. So it creates pores. These bubbles last 50 nanoseconds, but the membrane is perturbed during many seconds or even minutes because you perturb, this double layer membrane. And basically you can enter now some here. This is an example with the GFP, the green fluorescence protein here. So this is one example that we published many years ago, 2012. And actually, I started this project with someone at McGill, actually, Jean-Jacques Lebrun, who is the medicine department. You know Jean-Jacques Lebrun? No? Yeah. So I started this project. We were discussing but something completely different, and then we said, oh, yeah, you can do that, so why don't we try it? And it was successful. So the idea was to put here the breast cancer cell line in vitro, and then we put nanoparticles everywhere. They were not functionalized at that time, and just scanned here, and realized that this green fluorophore was after washing and everything. You follow all the steps, and then you arrive with the cells that looks like green when you do the fluorescence. And we did the transfection with the SMAD, the complementary DNA plasmid, and according to his result, the lipofection is not that great compared to the laser approach. This is in vitro.
Are the nanoparticles being taken up when you use that?
The nanoparticles are probably still around, but they are not broken.
Yeah, but are they getting into the cell?
No, they are just around. Maybe some are in and some are around, but they are not broken. There are some in. I don't have the statistics, but there are some in. What we did also, this is another paper, nanoscale in 2015. We put two cells. This is a proof of principle that we can do a co-culture, two types of cells that we were having in the lab. One is the breast cancer cell line. The other one is other line that actually it is related with the retina, but actually it is not. But anyway, this is a CD44 minus, CD44 plus. And then so we functionalize particle with the anti-CD44. And then do the scanning here and no scanning here. So this is a co-culture here. In blue, it's the nuclei. In green is the lucifer yellow untargeted cells that is going in, and in red, non-targeted cells. And the analysis of that is you have a selective optoporation. So laser is scanned everywhere here. It's a scanning that is everywhere, and basically you can include the let's see here, the lucifer yellow on targeted cells only, and non-targeted cells are not affected because the particles are not on the non-targeted cells. Okay? Okay. What else can we do? Some years ago, I had the idea of using this technique to stimulate neurons. So, and I was discussing with the people at Laval University. They are expert in nanophotonics. And finally, we have an interesting papers on that, an interesting paper. The idea here is to be able to in a neuronal network to stimulate with particles and laser and to stimulate action potential from a nanometer scale. Actually, is to open and close the channels on the cells. Maybe I don't know if some are familiar with the optogenetics. Optogenetics. This is something that is. It's not exactly the same thing, but it's using light. I mean, to perform some kind of transformation or reactions in the neuronal network. But we don't transfect the channels. It's the nanoparticles that are acting actually to open and close channels. So that's the idea here. The idea is to have a neuronal network, and with various types of particles, it can be various colors, various types, depending on composition and so on, and probably also various types of functionalization. And then you can have a nanometer scale stimulation and look at the effect. And the idea is to be able in the neuronal network to produce local heat, or to use extra charges, or maybe some kind of pressure on the dendrite, for instance, or maybe some kind of nanobubbles also, which is like a pressure wave on the local. And so we did that, and this is like this is one shot, and I mean the shot is at one place, and then what you see is the calcium that is getting in and it's become greener. There's a lot of details in the paper that I don't mention here, but basically we can stimulate the action potential in the neuronal network. So that's an idea that we'd like to pursue. If some people here have some ideas of pursuing that, this is pretty interesting. Right now, they are not doing that anymore in Quebec City, but something that we can do here in Montreal. I have the system for doing that. I don't have access to neurons, but neurons have to be specific. You have to take the brain of an animal to do the culture and do the experiments right away. Okay, another application. That application is, I have this technique, and I say, where can I use it in vivo? Where can I use it? Where can I use a laser in vivo? And the idea came out, and I had that idea for a very long time. The idea is in the eye. Of course, we would never say to people, don't shine a laser in your eye, of course. But ophthalmologists are doing that every day. I mean, they do a lot. And then they use that to do surgery in the eye. So the idea here is, I have a patent on that. To deliver biomolecules into the eye with that technique. And so this paper, nanoletters, that we show that we can do that in an animal model, in like a rodent, and to directly inject particles and then look directly into the retina. Ocular diseases affect millions of individuals worldwide. These diseases are either incurable or only transiently improved with current therapies. And the idea is to use high throughput, non-viral. There's some kind of limitation. A lot of people are using viruses, and it looks good. Biologists are looking for alternative way of not using viruses for different types of reasons. Personally, I'm not sure if I would like to have viruses in my body. They use like HIV or something like that they empty out. So the idea is to develop laser nanoparticle, like a virus-free technique. What we want is to deliver a small interfering RNA into the retina cell and to address eventually age-related macular degenerescence in the retina. And so this is the colleague of mine at Hospital Maisonneuve-Rosemont. They're doing a lot of study on this. This is a illness that is getting with age. I mean, when you get old, you start seeing something black at the center. So that's what we want to address. Okay. So this is our little friend now. So we use that. We did that in the hospital. Right now, we're starting to do that in our lab. We've been told through all the ethics. This is very time-consuming, by the way. So you're probably familiar with that if you're using animals. And then the idea is to irradiate at the good place and then look at the result. The first thing that we did is that after irradiation, and then we sacrifice the animal, and then we look carefully at the retina to see are the particles there. And basically, without going into much detail here, and with the technique that we have elaborated, so we take this piece and then we look, it very precisely, particles are at the retina level and not at the other level. This is like half a micron up or half a micron down. And so it's not stuck in the inner limiting membrane, which will not be very interesting. So the nanoparticles are at a good place. We are very lucky on that. It's because it depends where it is injected. It has to be injected at the good place. By the way, injecting things in the eye, this is something that is done very regularly by ophthalmologists. Just on a personal point is my wife is getting this injection almost every month now because she has a problem. She's getting injection, and it's a question of a few seconds it is done. Yeah, after that, I mean, she doesn't feel very well during 2 hours, but after that, it's okay. Okay, so we developed this optical, yes, I have this problem here, with this system here. So this is the in vivo laser-mediated silicon siRNA delivery. So this is only one spot. So because of limitation right now, we did not develop the engineering of scanning, but just, I mean, getting the laser at a good place. Here with confocal microscopy on the retina, this is, I mean, sacrifice animal, and then you look at under the microscope. And the nanoparticles were functionalized. SY5 is red, siRNA, and then we see the red after washing and everything. You see that the red is there. So this is the pretty good news. And then before I put forward the patent, I insisted that this was done without going into much detail about all the counting and so on. But one of the points that we did here is that 1 week after injection irradiation, we count the number of cells. And we realized that it is the same way that it is in the control, the intact or saline here. So non-toxic, non-toxic. So that's the point that I realized that this is pretty good. So we are right now on this project. I have a research grant from CRHR and NSERC to pursue that. And also, I am part of the nanomedicine research network in Canada, based in Vancouver, and we're adapting this to the cornea. And right now, I have a good part of my team that is developing this. It is. I know it's a very long way, but we started a very long way also, but it's a very long journey to arrive with a technique that will eventually work. Okay, so actually most laser cutting is like this. Probably in the future, therapy will be done with that. Okay, so I have more than a dozen papers. I did not cover all that in detail, but there's a lot of physics and application on this technique. This is some of the papers that were published. Okay, I'm switching topics now. Plasmonic nanoparticles for bioimaging. So let me put this here. Sorry for the people there, but they told me that I should not use my pointer, so I am close to the screen. This is silver and this is gold, and you can buy this. You can buy, $300. You have. We make them. We make them in the lab. And this is silver, and depending on the size, it's either transparent or more yellow, depending if it's bigger, it will be like this here. So there's plenty of space between these two. So we have this patent that we introduced that the people were able to make the gold silver particles of different colors like this, but it was small. But in application, I did it bigger. But when you started bigger, the size was pretty large. Size distribution was pretty large, and it was not very useful. So we have a way now of doing that. So with this pattern and published with in many papers on that, this is the technique is described here. So as you see here, this is 56 nanometer plus or minus 10%. And this is pretty good. This is called, fine distribution. This is the best thing, the best you can have, actually. So the idea is that we can have nanoparticles of different colors, and we can functionalize them with this way. This is an example that with the various biochemistry steps that we published that in nano letters. So the idea is the following: you have different colors, and then you can functionalize differently depending on the color, and then you put that on cells, and then you look at it with different optical setup. And this is a reconstruct image where different colors that are calculated depending of the spectra. So the spectra, the imaging like that, it's called hyperspectral imaging. So this is like doing X, Y, Z, lambda. So four dimension. This is hyperspectral. So that's the idea. The idea is to use this. So a lot of people are in biology, and there's probably some of the people here, they use fluorophores to color different parts of cells so that you can see what it is, and different types of fluorophore, different colors. Usually these fluorophores, or most of them, they have photobleaching. Photobleaching is part of them in fluorophore. Fluorophore, when you irradiate, they change conformation, and at some point, the intensity is changing. It's difficult to make with fluorophore to make quantification. The pathologists that I'm talking with, because we're collaborating with the pathologists on that, the fluorophores, you can do subtype of quantification, but it's qualitative. They want to have something that is quantitative. What we're trying to develop right now is to use, in addition to the fluorophore that the pathologists are using, we want to use this type of nanoparticles that would be functionalized, and then we can see them, and also we can count them. So counting is important. So that's the idea. So we have optical setup that is developed, different types of colors, and different types of way of irradiation and collecting. This is the optical engineering part, but basically this is the conventional dark field, which looks like that, and this is our approach, which is much better. You still see the cells, but also maybe the image is not that clear, but you see also the nanoparticles. And this is another setup that you can do X, Y, Z, and also lambda. And this is a reconstruction of this of the image here. So this is also an identification. So with the idea of that, we start up a company, Vega Photon, which is making the optical setup and also the use of nanoparticles. And so if some people would like to use our system, you just contact me, or mostly it's my research associate that is in charge of building all this. So we develop imaging and detection technology based on the plasmonic optical properties, though these nanomaterial, and we essentially produce systems for measuring some of the properties, as well as looking at the optical properties of samples. And this is the one that I think is the most interesting one for the moment. We're developing also a new camera, hyperspectral camera. So this is little video. See, here, this is a conventional microscope. And so this is a setup where you can have different colors that will interact with these different colors there. So basically, you can put your sample, and the pathologist, my collaborator, she really likes that because you don't change what she's doing. She's just putting that there, okay? And basically, you can see, like here, 3 types of nanoparticles, and depending on the way that we are irradiating, we can see the various colors, and this is only like the silver. Silver is like blue in scattering, by the way. So it's, I can explain that in question if you want. So this is a dark field image that is a conventional way, and this is the use of our, it's the same thing. It's just that we put the light on the side, and what we see is the cells, but now with all the various nanomaterials, and then we can play with the Z direction to see where these nanomaterials are. So this is a cost-effective side illumination dark field. So it looks like that. Okay, this is not a galaxy very far with stars. They are cancer cells with nanoparticles. And so we can detect now 4 types of nanoparticles on cancer cells, and you can do some image analysis to arrive to that. A project that we started, but right now it is interrupted. This professor at St. Justin is working on completely something else. But if there are some people interested in this aspect, I found the project very interesting. This is the retinoblastoma. This is the cancer in the eye of children. It is not very common, 1 out of 20,000 births. But however, if it is your children, is very important. So this cancer is in the eye, and then if it's not removed, it will progress and then get to the brain, and then this is the catastrophe. So the idea here is to do that. Of course, we didn't do that on children, not an animal at this point, but we started to work on what it's called like a phantom, so try to simulate what it is. So we have here like cancer cells in suspension, and then we put nanoparticles. And what you see here is the cells with gold nanoparticles. You have gold nanoparticles everywhere on the cells that are in suspension in this. What is this? This is to simulate the vitreous here. So this is like a Z scan, as you see here. This is like a cancer agglomerate, and then you have nanoparticles everywhere, and so maybe just to have it in another time. Is it running? Yeah. So what you see in the Z-scan, you see all the various nanoparticles inside. So what you can do, I mean, you irradiate and then you kill the cancer cell. So we did that. This is the in vitro approach to show that in these conditions with this type of laser that we can, this is not femto, this is more a conventional Nd:YAG laser, 80% dead cells for this primary study. So these are other papers on that. So as a conclusion, probably my time is out. I presented biomedical applications of nanophotonics and ultrafast laser. We can do 3D multiplex bioimaging, application to pathology. We can target nanosurgery for cell transfection, drug delivery, gene therapy, neuron stimulation, application in ophthalmology and also in cancer therapy. It's a picture of my group during the beginning of the summer, looking for a good PhD student that would like to couple physics, engineering, and medical application with people also in a hospital. So there are around me some research associate postdocs, students in biomedical engineering, and student in physics. She is in physics and he is in physics there. He is in physics. Some are more on bio. So it's a mixed team. that's pretty important. So I'm looking for students because all the projects I have money. I'm looking for people. So that's so for my research group, yeah, people interested in working with laser, plasmonics, biomedical, as well as also animal studies, so more on the bio side. And if I come back and put my hat of chair of an engineering physics department, maybe this is not maybe the good audience, but anyway, there will be several tenure track openings in the near future. This is physics physics background here. So I would like to stop here, and I would like to thank you for your attention, and I'm open for question and discussion and so on. I think there's a half an hour discussion, or yes, yeah.
Whatever it takes.
No, not everything. Okay, thank you.
Okay, so questions or rather clarifications?
So that's gorgeous talk.
You have to be
Very nice
short.
talk, but it seems that the major problem is still going to be to get these nanoparticles in the right place. And presumably also, if you want to use these things in vivo, you need to be able to get the light, the laser there.
Yeah.
So have you thought about both of those problems?
The solution, I mean, as I presented, this is in ophthalmology. So we can inject nanoparticles and we can get the laser there. So in ophthalmology, I think these two problems that I taught at the beginning, I mean, this technology can be applied to that. And we have a proof of principle, at least on animals, and then we're pursuing on that. So yes, it is a real problem. Technique does not solve all the problems of life. It's very specific. So I need particles at a specific place. And also I need the light at a specific place, yes. In vivo. In vitro, I can do many things. I can have, I mean, it's a question of throughput. I have to scan, I have yeah, there's some of my colleagues in Germany, they have done some work on that. They're trying to get high throughput. I mean, cells that are flowing in microfluidics. And then you put nanoparticles, and then you scan the laser. You can do this. You can do this transfection on many cells.
So, for example, I guess that would mean that you could think about treating blood cells or something like that.
Yes, in principle, yes. I did not work on that. There are some other people working on that. I did not work on that. I'm concentrating now on the eye, which is a very difficult problem in itself. So. But neurons also, I think it's interesting.
You mentioned that you make these nanoparticles. I was just wondering how you actually go about doing that.
How do you, what?
How do you make the nanoparticles in the tubes here?
Okay. You create with some kind of, you start with some chemistry. Faraday was doing that 150 years ago. So you put a salt of gold, and you transform your gold that is in a boiling water. You transform your gold that is plus in gold zero. So you add up a nitrate that will do that. And when you have gold atom, they start to form particles. And then you have to do that very abruptly, and then it grows. So you create seeds, and then it grows. So this is chemistry. It's basically, this is chemistry. You can almost do that at home. If you can handle, I mean, and you have boiling water. Yeah, I can show you a video. Usually I can show a video of that. I mean, it takes 4 minutes. 4 minutes to make that. To make this, it's longer because what we do, and our patent is based on, we start with a seed, and then we add up something on it, and then we stop, and then we add up another one, and then we stop, and then we add up another one. The problem is when you add up, you don't want to create new ones. So you have to control what you're producing. And because you don't want to have a distribution that is very fine. If you start having, growing one nanoparticle and the other one is a new one, you will have two distribution. So you don't want this one, you just want this one. So you have to control the condition. But this is what we are doing, and right now it's pretty good. So we can control this.
Bit of a different question. In the earlier stages of your presentation, you're showing that if you use nanoparticles, you could irradiate with an unfocused field, basically, and that will create pores because of the energy concentration around the nanoparticle. And my intuition is that perhaps these nanoparticles don't have to be spherical; they could be of any shapes, more or less. Does that change the constraints on the kind of the focusedness of your field, or do you have to adjust that to balance out different concentrations?
That's a very good question. I don't know if you're familiar with the field. Are you talking about nanorods? Are you talking about that? Or?
Not specifically, but you had shown like a rod, right?
I've shown at the beginning nanorods. It's
right,
because
like
I have
quick.
this. I could have shown, nanoparticles, just that I'm using that figure, which is pretty nice. The whole is like a little rod, yes. Nanorods can be made. You can buy them. We have used them and realized that they are not that good. Actually, nanorods will tend to have a peak, the plasmon peak here. I will go in much detail now to answer the question. The plasmon peak here is around 520 or 530 nanometer. For the nanorod, it shifts to the near infrared. We can have nanorods at 800 nanometer. If we take a nanorod and we irradiate at 800 nanometer, it will absorb a lot. If it absorbs a lot, it will destroy the nanorod. I don't want to destroy the nanorod. If I use an energy that is smaller, the nanorods will heat up a little bit. And a nanorod that is heated up will no be longer a nanorod. It will transform as a sphere. Nanorod is not the minimum energy. It's the sphere that is the minimum energy in terms of energy of the material. So basically, nanorods are not stable. So they are not useful. So yes, we can play with shapes, but the best one is this one.
So for a given material, different shapes will have different absorption
Yeah, but we mostly be talking here about, because we need this near field, we talk about metals, and metals would be mostly silver, gold, maybe a little bit of copper. Some people have used maybe aluminum or some other types of plastic. It's called plasmonic materials.
Thank you.
You need that. It's more like the physics of plasmons.
I have two questions regarding your experiments with retinoblastoma and hyperthermia.
The retinoblastoma? Okay.
Yes. First of all, what size of nanospheres are you using?
In this case, it was, as a proof of principle, we use what we have, which sometimes more standard is around 100 nanometer. It can be smaller. In this process, it's basically, it is absorption. And absorption goes with the volume. Bigger the volume, larger is the absorption, the heat, diffusion. And so in this case, as a proof of principle, we use 100 nanometer. But sometimes we use smaller. It depends on the combination with the types of laser, wavelength, size, shape. Exactly. This is all the engineering development to arrive to the final result. But basically here, this is not particularly new. The nanosurgery I'm developing with the femto, and I'm probably the most advanced group on that. Using nanoparticles to heat up and kill cancer cells, there are many groups in the world doing that.
Okay.
A lot of groups. There are some groups that are close to go in. Noami Alas, she's pretty close to have in a clinical trial over number 2 or number 3. Which is quite advanced in terms of going to human. This is human study.
Okay. And my second question is if you have experienced any thermoresistance effects when you are heating the cancer cells.
Thermolysis effect?
Resistance, yeah. Cancer cells can adapt to increased temperature. And they...
It can adapt to increased temperature.
Yeah, and they do not die at some point.
Yeah, but with that, we can increase temperature very high. So at some point, it will be killed. You talk about, I mean, the temperature that is slightly elevated, but not too much.
Yes, apoptotic levels. Yeah.
Oh, yeah, but I can with that. I mean, with the laser and nanoparticle, I can melt the nanoparticles at 600 or Celsius. It can be, you can go high temperature. The point is, I mean, again, just, of course, is the combination with the laser and penetration and all that stuff. But basically, adaptation, I would not see that. I mean, we just increase the. It's proportional. So we increase. I don't know if we have an increase from body temperature to, let's say, 50 degrees or we can go to 80 degrees. We just increase a little bit the laser, and that's all. Or we can go much higher than that. I can reach thousands of degrees easily. Yes.
I have a question regarding the multiplexing. Is for the max of colors you can separate, or can you go beyond 4 different nanoparticles for that approach?
Yeah, we're trying to have more nanoparticles right now. Right now, we're showing with 4. There are 4 types of nanoparticles, and I think we can go more. Because we're using our irradiation, that the small device that I show, it's very small, convenient. You put it there and you have your 4 nanoparticles. It's pretty easy, but the point is that we can go further by developing with the hyperspectral. This is what we are doing now. So number can be increased, but not as much as, let's say, I don't know, quantum dots or something like that. Quantum dots, of course, you can have, I mean, much finer, like fluorescence. It's like your fluorescence. You irradiate, and then you get your signal. But this is an overall photoluminescence system. Right. I'm doing exactly the same type that what the pathologists are using, basically.
So you showed that for fixed cells, if I understood your data correctly. Can you do it on live cells?
This is a very interesting question, and this is what we are doing. So yes, actually, part of the project we're going to publish in few weeks. So we're finishing the paper. We're trying. It's not that easy. Like cells in suspension, like. Let's say my pathologist is telling me that if you take, let's say, with fine needle aspiration, you get your blood or whatever through the, let's say, from the poumon, lung, the lung, and then you do you put the various types of nanoparticles. We are doing this on live cells and then fix them after. It's a challenge, but this is something that we have in mind of doing. So we have a written grant proposal, and essentially our introduction is exactly that. But right now we're working on fixed cells. But we have some results. It's encouraging, but still to develop.
Hi, very interesting work. Thank you for coming and sharing it with us. I have a question regarding the cavitation of the nanoparticles. When you apply it in vivo, are you worried that you want to keep these cells alive for gene transfection? Are you worried that when you create those pores in the cells, you'd have an accumulation of nanoparticles inside the cells that would eventually, over time, become toxic to the cells?
That's a question, that's a very good question that we have to test. Right now, the test that we have done is we've done the test, and then we waited 1 week and look what happened. Right now, I have one PhD student that is pursuing in this direction. So the idea now is to carefully create AMD, age macular degeneration, and we create that with a laser, actually. This is very strange. And then we try to solve it. We try to do our technique and try to see in time what is happening. And yes, we have to look at this. I cannot answer. Of course, I mean, you may have some kind of side effects that may be a longer time. Any type of treatment have effects on a longer time. That's basic problem of treatment. It's like chemotherapy. You have a lot of problems, potential problems.
Just as a follow-up question, you use the targeting moieties to target
targeting what?
Moieties. So specific. Yeah. So you target the surface of the cells. But have you looked at targeting intracellular compartments like organelles or stuff like that?
In principle, no. The answer is no, and probably it may be not really appropriate for our approach for that. It doesn't go inside. We have to probably to use particles that are smaller, and then through endocytosis get inside and so on. It's much longer. It's more complicated process, though. I would not see too much hope in that direction for that.
Any other question? Actually, I have one. So things are happening at the, oops, sorry. Okay, things are happening at the nanosecond pulses, and they are very different at femtosecond. Nanosecond is 10 to minus 9, femtosecond 10 to minus 15. There is a lot in between. Do we have a threshold when things are, I know that the price of a nanosecond laser is way lower, exponentially lower than the femtosecond laser, but other processes where they suddenly change to very sharp cuts and things like that?
You're talking about the physics about what's going on around nanoparticles and liquid and irradiating and all the various phenomena. We look at that and all the details in many of the papers that we publish, actually. So the question is, do we need a femtosecond laser? This is what we have. Right now, we're doing some work using, this is not published, using picosecond laser and playing with the wavelength. It looks like it's pretty interesting also. Probably the process is different. The process I'm explaining is due to the near field amplification, and then there is this all the various types of, steps of processes occurring. If it is picosecond, probably the same thing. If it arrives with nanosecond, we have tried nanosecond, it's a heating process. So we need to be as much as possible in resonance, so to be at the peak. So a particle like this absorbs at 520. So now if you're in a biological media, 520, a lot of things absorb. So it's not the good way to go. So there's a trade-off. Or we shift this to the near infrared with the core shell structure. So you can have core shell structure that shift to the near infrared. This is less stable. So your question is, how can we explore the various combination of laser, wavelength, nanoparticles? So the answer is yes, there's a lot of things that can be done. In practice, yes, limited amount of, I have a lot of laser, but there are some laser I don't have.
Any other question? Okay, thank you. Thank you.
Thank you. Thank you.